Good afternoon. Greeting from Stockholm. My name is Ruoli Wan, Assistant Professor in Biomechanics from Royal Institute of Technology. It is my great honor to chair this research breakout session in biomedical digitalization. During this session, we will have 4 exciting talks covering a broad perspective of digital transformation in biomedical applications. The Q&A session will be after all the talks. If you have any questions during the presentation, I encourage all to leave your name, the person you would like to ask, and your question in the chat window. I will moderate the questions after the talk. Speakers will have the chance to answer your questions live. Without further ado, I would like to welcome the first speaker, Seshagopalan Muralidharan. From KTH, he will talk about digital DNA diagnostics.
Hello, everyone. Welcome to our presentation for Digitalize in Stockholm 2021. Our project is titled Democratizing DNA Diagnostics. The research on our project is being conducted by the principal investigators Mahir Hamedi and Björn Muller. Also on this project are Pedro Rio Carvalho and me, Seshagopalan. Let's typically look into how sample collection and testing is done for, say, the COVID-19 infection. I know a lot of you must be familiar with this, but let's just go over this in really quickly. What happens is typically there's a swab that's taken, either through your nose or through your mouth, and then they extract the RNA, and then this is converted into DNA. Once this extraction is done, your sample is sent to a centralized testing laboratory where the testing is conducted, and then you get the results. The key word here to note is that this kind of testing is done in a centralized laboratory, and you don't have access to these kinds of testing tools. We do have access to some testing, but this kind of testing is typically not very accurate. For the COVID-19 virus, which is typically an RNA-based virus, a conversion to DNA is done. Once this conversion to DNA is done, the DNA molecule is taken, it's split apart, and then it's multiplied many times until you get billions of copies. In this case, when you have 40 cycles, you end up with nearly 1,000 billion copies. And this is the true power of RT-PCR testing, which is the fact that you start off with a very small sample, but then you amplify this and then you get a large sample. So any testing that has to be accurate will have to sort of incorporate this process of amplification. Typically, when COVID-19 testing is done, amplification and detection of the virus are done in the same machine. And the way that amplification is done is through a process called thermocycling. So in thermocycling, you start off with a low temperature, which is in this case would be room temperature. You go up to 94 degrees Celsius where you denature the DNA, and then you perform, and then you add these molecules, which will basically recombine and then create new DNA molecules. You repeat this many times, and then you complete your PCR. This form of amplification is typically done in a PCR tube, which is also called an Eppendorf tube. And these tubes are actually placed into large machines like the one that you see to the right. And these machines typically hold between 90 to 100 tubes, and all of these can be tested at once. This machine also does amplification and detection. So for detection, there's basically a light that's sent into the sample. You calculate the reflected wavelength, and based on that, you try and detect if the person is COVID positive or negative. In our case, we will be using electrochemical detection, where instead of light, we use electron transfer to determine if the person is positive for a specific part of the DNA that we're testing for. In this case, this could also extend to cancer detection or even Ebola detections. For our project, we've sort of identified a loose set of requirements, which is that our project is low profile, it costs very little, and it's simple to design, it's accurate, repeatable, and it would also be nice to power this from a standard phone charger, and also if it would come with Wi-Fi and Bluetooth connectivity. So considering all of this, we had to first develop the thermocycler where this amplification is done, and then we also had to develop the associated electronics that go with it. So the thermocycler, where the amplification takes place in this case, is basically just this dome-shaped thing, which is containing a slot in a PCB where the sample would be placed. This sample would be heated through built-in heaters on the PCB. The advantage with these heaters is that these heaters would also serve as temperature measurement devices, hence ensuring that we get very accurate temperatures in the PCB. For cooling, we will of course have an external fan. Given this, we also have additional external electronics, which will be used to sort of control the reaction that takes place inside here. And this is the board that we've manufactured. This is a rendering of the board. As you can see here, you will notice that we have a fan, we have a controller unit, and we have various other electronics that go with this. Once this was built, we realized that the cost of this is quite low. The cost of consumables in this case is as low as $3, which would just have to be the PCB, a gasket, and a top covering lid. And we have some one-time electronics, which in this case is the electronics that's actually heating, which is actually creating heating in this thermocycler, and also for the control electronics. And this is estimated to be around $40. Now that we've done this, our future work will be in testing the PCR in this PCB amplifier that we've built. We're going to be integrating electrochemical detection into this PCB. We will be testing these, we will do our final development, and then we will release our hardware design and software. We're going to make it open source. Thank you. These are our project members.
Thank you, Sashan Gapalon, for a very interesting talk. Let's move on to the next speaker, Rodrigo Moreno, also from KTH. The topic of the presentation is about mechanical tissue properties of the brain.
Thank you. Our project is called Characterization of the Mechanical Tissue Properties of the Brain in the Developing Brain with Magnetic Resonance Elastography. And this is a collaboration between Armita Goldkar from Stockholm University and myself, Rodrigo Moreno, from KTH. The brain is developing from birth to young adulthood. However, in this project, we are interested in this time window between childhood and adolescence. And this is because the brain has a dramatic neural development during adolescence. And adolescence represents the peak age of onset for anxiety disorders. And changes of mechanical properties of the brain have not been assessed before, and we wonder if these changes are related to the risk of developing anxiety disorders. In this project, we will have the following approach. So we are currently recruiting two groups of children. One is from 9 to 11 years old, the other from 13 to 15 years old. These are recruited in schools in Stockholm. And we are going to use MRI, magnetic resonance imaging, specifically two types of MRI. One is MR elastography and brain connectivity, as you can see here. And then our idea is to use these sequences to assess changes in the brain in these two groups. Also, we are going to run some questionnaires to get some predictors of anxiety in these two groups. And our idea is to try to find correlations between imaging findings with these predictors that are obtained with the questionnaires. One of the main tools in this project is magnetic resonance elastography. So the aim of this method is to obtain mechanical properties of the brain through MRI. How it works is that the subject is in the scanner, like here, and then we use a pneumatic driver to induce some kind of waves into the tissue, in this case in the brain. So then these waves are propagating in the tissue, and then you use MRI for acquiring this wave propagation. Once you have acquired these images, you can use some post-processing to reconstruct the mechanical properties of the brain, as you can see here. And right now, KTH is the only site in Sweden with this technology. MRI reconstruction is challenging, and has been shown that artificial intelligence is promising for solving this problem. So the current methods, what they do is to generate synthetic mechanical properties of the brain. And then they use that to generate synthetic displacement waves, synthetic MRE. And then they use these synthetic images to train a neural network that is able to predict the original synthetic mechanical properties. The main problem of this method is that it's not is only using synthetic data. So in this project, we're going to use real data acquired in our scanner in a second step. So the first step is more or less the same as the previous method that I described. But in a second step, we're going to add the acquired MRE images to get better estimations that consider the particularities of the MRI scanner. That usually it's something that you have to consider. Also, we are going to use a method that is called physics-informed neural networks, where PDE solvers, numerical methods, traditional numerical methods to solve these problems, are combined with machine learning to get the best of the two worlds. Thank you for listening. So this is us, and we are currently recruiting a postdoc for this. And this is in collaboration with Philips, that our MRI scanner is from Philips, and also with Mayo Clinic that will provide us with the hardware that is required for this project. Thank you. I'm happy to answer questions.
Thank you, Rodrigo, for a great talk. The next presentation is from myself and my co-PI, Yuanyuan Li. Good afternoon. My name is Rolly Wang from Royal Institute of Technology. Together with my co-PI, Yuanyuan Li, we represent our digital pair project, aiming to use digital tools to facilitate personalized medicine in rehabilitation. Impaired motor function is one of the major disabilities. Person may lose their ability to perform daily activities and largely influence health-related quality of life. The management of complex disability currently largely rely on rehabilitation. However, supervision and assessment of a rehabilitation pattern is still a medical and engineering challenge due to the fact lack of biofeedback information about the effect of the rehabilitation motion on the individual human biological tissues and structures. Therefore, the project aims to develop the digital twins of the personalized human neuromuscular skeleton framework. We allow carrying out a high fidelity real-time analysis of non-measurable internal biomechanics quantities in a non-invasive manner. As a proof of concept, we proposed a clinical-friendly DT framework that provides a real-time biofeedback to facilitate rehabilitation training and intervention. Computational neuromuscular biomechanics has been applied mostly in research setting, aimed to better understand various motor disorders and facilitate rehabilitation technology development. However, to translate the power of a computational model from a research setting to a clinical environment, we face several major challenges. First of all, the model requires high level of personalization with respect to anatomical, physiological, and neurological characteristics. Second, most skin contact sensors suffer from limited signal stability and discomfort during long recording. At last but not least, data input still largely rely on lab environment, for instance, a marker-based camera system. We will target these challenges in three perspectives in this project. First, to develop innovative electrodes which can record biological electrical signals and ultrasound signals simultaneously. This can largely simplify the calibration procedure. Yuanyuan will talk more about our innovative sensor idea a bit later. Second, the proposed DT framework will incorporate data-based and physics-based modeling approaches, which will increase the reliability and fidelity of the parameter estimation. Also, we will use variable sensors to achieve the goal to be feasible to apply the framework in a clinical environment.
To simultaneously collect the electrical and ultrasound signals, one can place metal electrodes and ultrasound probes close to each other, but this is challenging for small muscles. Another approach is to embed metal wire electrode into silicon substrate. However, the metal wire electrode is not ultrasound transparent. This will influence the ultrasound signals collection. In addition, silicon substrate is not sustainable since it is from fossil-based resources. Our strategy is to combine the biomass-based adhesive substrates with biocompatible ultrasound transparent electrode. Then through printing technology, like 2D printing or 3D printing, we can make ultrasonic electrodes. To make biomass-based adhesive substrate, we first extract nanocellulose from wood and then form this ultrasound transparent substrate. Regarding the biocompatible ultrasound transparent electrode, we plan to use isographene or P.PSS. Then using printing technology, 2D printing or 3D printing, we can make ultrasonic electrodes or ultrasonic electrode arrays. This project is a multidisciplinary collaboration between the fields of biomass-based advanced materials design and computational biomechanics of human movement. Through this project, we want to provide a state-of-the-art, feasible and reliable digital tool in personalized medicine in rehabilitation towards healthy life. Thanks for listening. And we also want to acknowledge Digital Futures for the funding and the event arranged.
Let's move on to our last talk of the session, given by Erika Zelio, also from KTH. She will talk about the biodegradable components for packaging.
Hello everyone, my name is Erika Zeglio from the Division of Micro and Nanosystem, and today I'm going to present a project in collaboration with Tiziana Fuoco at the Division of Fiber and Polymer Technology. So when we look at food waste from Sweden, for example, we see that most waste is produced in the households. But also from other steps all the way from production to consumption. A smart packaging solution have been introduced to monitor food all the way from production to consumption. And from producer, it offers the advantage to have real-time and close to production monitoring of food products. But for consumers, it also provides the possibility to have an alert system over food spoilage and to reduce waste. However, solutions to date are not biodegradable. And for packaging, biodegradable polymers have been introduced to reduce plastic waste. However, for electronic devices, we have only a few examples of water-soluble electronics that have been introduced mostly in biomedical devices. So future biodegradable technology will need to basically have the stability that is required for application in humid or moisture-rich environment, but also be degraded at the end of their life cycle. And we foresee that this is going to happen through enzymatic degradation. So our vision is basically to use enzymatic degradation to develop biodegradable labels for food monitoring. And in this project, we really aim to combine our reciprocal expertise. Tiziana Fuoco is an expert in polymer chemistry and synthesis, and especially in designing polymers with specific degradation behavior, but also on methods to monitor degradation of polymers. On my side, I am an expert in devices for organic electronics and bioelectronics, and particularly in a class of device called organic electrochemical transistors, which can be used both for amplifying small biological signals, but also for sensing. And in this project, we really merge our expertise in biodegradable material and device engineering to push toward technology for packaging digitalization that are also good for the environment. And basically, our view is to generate a sensor who will merge, on one hand, control biodegradation and also the possibility to integrate into life and the environment, but that also has high performance and stable operation need for monitor food products, for example. And the substrate is going to be composed of polymer that can be degraded by hydrolyzing enzymes, while the core component is going to be a conjugated polymer, which will have both the stability needed for operation in contact with aqueous electrolytes, but also enzymatic degradation. Also, the contacts are going to be made by iron, so they're going to be dissolvable, and their dissolution is going to be regulated by the substrate material, who is going to be used also for encapsulation. And our project, if in one hand we are going to have polymer chemistry from synthesis to material degradation, on the other hand we are going to work on the microfabrication and testing of device components. And merging this expertise will enable us to produce, on one hand, biodegradable devices, but on the other hand we are going to integrate some sensing elements needed for food monitoring. So to have both biodegradation and sensing capabilities. And this project is just at the beginning. We basically didn't start it yet, and but we hope that it's going to be the beginning of a long-term endeavor and collaboration in order to merge digitalization and sustainability. And I hope you enjoyed this short presentation, and thank you very much for listening.